thank you very much for the introduction. And I would also like to thank the organizers for inviting me back again to Singapore. And it's always a great pleasure to be here and to enjoy the environment and to talk to the students of this meeting. As said, I work at the Max Planck Institute of Biophysics in Frankfurt, and we focus to work on membrane proteins because we think that membrane proteins are the most important things to study, and without membrane proteins you cannot think. But I'm not going to talk an individual membrane protein, but rather I try to talk about projects which we need and challenges in order to understand ourselves as good as possible. And so we have to understand ourselves, and our body consists of 37.2 trillion cells, and the cells form tissues, and tissues from organs, and the organs from us. And you see here a number of the organs which constitute our body. And the most important is to understand our brain. And only if we understand our brain, we can understand how much we can understand. And you see here, for instance, Broca's area, which is the production of speech, and I hope it's active in my brain. And this is Wernicke's area, which is needed for understanding speech, and I hope this is now active in your brains. And so most important thing which we had recently was the sequencing of the human genome, and we have about 3.1 billion base pairs on 23 chromosomes. And of course, you know that 3 bases code for an amino acid, the amino acids code for proteins, constitute the proteins. And we also have to know the number of protein encoding genes. It's not known with certainty. It has been believed to be 23,000. Now it has been reduced to about 20,000. And now the telomere to telomere sequencing suggested the presence of 19,969 protein encoding genes without the Y chromosomes. And proteins are encoded by only 1.5% of the genome. The Human Protein Organization lists 19,778 protein encoding genes, and people have tried to verify them experimentally, but they still miss about 7% of the proteins which are suggested by the genes. The recent addition is the complete sequence of the Y chromosome, and it added 41 new protein coding genes. So males have 41 genes more than women. Now our task will be to identify all protein encoding genes and the non-coding RNAs which regulate the gene expression, and we have to find the proteins and determine their function. And when we know this, we can study the disease relation of the protein and develop therapies. We can learn from the genomes, which are 99.9% identical between all humans, and you can identify genetic diseases. You can make deductions on metabolism of medical drugs. Some people cannot metabolize one or the other drug, and you should know that from the beginning. And you also get information about transplantation antigens, which are required for identifying suited organ donors. And my proposal is it's very important to sequence the genome of each person after birth or even before. And from the correlation of human diseases and respective genomes, we can determine the risk for genetic diseases, and an individual then can change his or her lifestyle accordingly and minimize the risks. And now using artificial intelligence, there are new correlations are discovered. For instance, use of machine learning to identify mutational patterns in 60 genes identifies 60 genes that increase the risk of abdominal aortic aneurysms. So very important. For that, we need the bioinformatics people, and we have to identify all possible correlations. I think we should start for a complete understanding of ourselves with the fertilization of the egg by the sperm, and here's a basic picture how this happens. Next is that we have the cell divisions to 2, 4, 8, 16, and 32 stages. And the 32 stage, you find a differentiation into various cell types, whereas before that, all the cells are more or less identical. And so we have to understand the differentiation into the various cell types. We have to understand the formation of tissues. The tissues form the organs, and the organs from us. There's a very important development is the creation of organoids. Organoids are three-dimensional versions of organs that are derived from stem cells. And they can self-organize and differentiate into three-dimensional cell masses. Very important now for studying and identifying drugs for treating diseases which are related to these organs. The next steps are then, there are more than 500 different cell types, and the number increases steadily. And the Human Cell Atlas is a global project to describe all cell types in the human body. And it's important to know that of the about 20,000 protein encoding genes, only about 9,000 to 10,000 are expressed in each cell type. And we have to know which proteins are present in each cell type, and we also need to know where they are in the cell. That's a picture of a typical cell of the human body. So in blue you see the nucleus, the nuclear membrane with some holes in it, which is used for the export of RNA. Then you have mitochondria, the power stations, you have the waste degradation stations, the lysosomes, you have chemical factories like the Golgi apparatus, and you have the cytoplasmic membrane. And the cytoplasmic membrane is the most important thing for drugs, because most drugs to cure diseases act on membrane proteins at the cytoplasmic membranes. And the Human Cell Atlas actually catalogs all cell types, and also it maps the cell types to the location of the tissues and within the body. It distinguishes cell types, and it captures the key characteristics of cells during transitions, such as the activation of the cells, and it can also trace the history of cells through a lineage within the human body. And the results serve as a basis for both understanding human health and diagnosing, monitoring, and treating disease. So now we need the methods which we can use. So basically we have the genome by use for DNA sequencing. We have the transcriptome, that's the messenger RNA, which we have to determine. And we have the proteome, which is the most important thing because the proteins are doing the job, the function at the end. When we come to the so-called now the Omics world, we have the genomics, that's straightforward. We have the transcriptomics, that's doable. And we have the proteomics. Proteomics is the most difficult thing, and it's the least mature in technology. So you still need further development. And quite interesting, Nature has called spatial proteomics the method of the year 2024. Here they use tissues, and the tissues you can differentiate the cells. And with the laser dissection, you can analyze, you can isolate the cells and can analyze them specifically and look for the differences and also make investigations of pathological stages. So that's a very good method. So we have the genomics, and genomics is very important for identification of mutations in cancer cells, because you need the driver mutation in order to identify the best therapy. Transcriptomics is doable even at the single cell level, and it's very important for the discovery and the characterization of cell types. Proteomics, as I said, most difficult, not possible with single cells, and the method of choice there is mass spectrometry. And there's one problem, that the messenger RNA levels from transcriptomics and proteomics, the protein levels do not correlate, because there is regulation of translation by proteins and by other RNAs. So this still has to do. Here's an example of an analysis of a human proteome of an HeLa cell, a cancer cell. And there are actually 9,667 human proteins have been quantified, and the reproducibility of these experiments are excellent. And the next task which you have is the identification of the function of each protein. And for that, we can use computational biology, which derives information from amino acid sequences and makes comparisons with related proteins of known functions. Experimentally, we can delete or downregulate the gene and look what happens in the cell. And the biochemists like to purify the protein and do detailed investigations by various methods of the protein. And then, of course, if we know the function, we will have information about the disease relation. Genetics helps, and there's one example is that genome-wide association studies have identified 40 targets, and actually 40 targets have been already used, and there are inhibitors in order to treat diseases. The prime example are the PCSK9 inhibitors, which lower the LDL cholesterol blood level to fight heart attack and stroke much more efficiently than statins and other drugs which are used currently. And it took only 12 years between genetic observation and the FDA approval of a PCSK9 inhibitor. However, there are many misannotations of protein functions. There's one example. We have studied the transporter SLC49A1 and the A2 transporter, which have been annotated to be heme transporters. And here you see the structure of a heme group. But actually, we found out that it actually transports choline and ethylamine. So this and not heme as was predicted. And so we did the structure determination by single particle electron microscopy. So that is the experimental result of the density of the protein. That's the method, the model which you put in. And you also can here see the binding site for the choline in great detail. So there is cationic pi interaction between the tryptophan residue and the positive charged part of the choline residue here. So we get information about the function of the protein also from our structural studies. And once we know the relation of the disease relation of protein, we can start to develop drugs either inhibiting or activating an incorrectly functioning protein. And the best way for that is drug design, which is based on known structures of the protein. And the methods for structure determinations are just listed here. It's X-ray crystallography, which was used by ourselves. You use mainly synchrotrons, costing about $200 million US. And the free electron layers is about 1 to 2 billion US. Next method is nuclear magnetic resonance spectroscopy, and the 1.2 gigahertz machines cost about $20 million US. And nowadays we use primarily single particle electron cryomicroscopy, and each of these microscopes costs about $5 million US. And what I would like very much is neutron crystallography, because you can identify the position of protons, and which is very important. And for that you need nuclear reactors or spallation sources, and these ones cost about $2 billion US. So we need international collaborations in order to build this and to run these expensive machines, and these are run by physicists. And of course, our task will be to determine the structures of all human proteins and of the structural changes during their action. So the recent hype has been, of course, AlphaFold, artificial intelligence. Last year's Nobel Prize in chemistry was awarded to AlphaFold. So we also try that, and of course, if AlphaFold would be perfect, we could give up our structural work and retire. But we have obvious problems with membrane proteins transporting large substrates across membranes. So I will show you here one of these examples here. For instance, we work also on the cytochrome B deoxidase from Mycobacterium tuberculosis in order at the end to be able to develop drugs for fighting tuberculosis. And here AlphaFold makes a very good prediction. Even in the absence of the 3 heme groups which are there, you get the same structure. So very telling you that AlphaFold doesn't take care of voids in the proteins, which I think is a little bit not well to be understood. And we also did the structure of an ABC transporter, so-called ABC transporter, which is a true heme exporter. And what you see here is that the predicted structure with and without the heme is shown here. They are identical, but when we do the experimental structures, they differ. Here you see the membrane part here. You see the structures derived from AlphaFold, the model from AlphaFold, and the blue is the experimental structure, and there's really substantial deviation from between the experimental structure and the true structure. And when you add the heme group, there's no change in the AlphaFold structure, but in the experimental structure, you get a much tighter binding of the binding sites, so the helices move together, which is not found by AlphaFold. So this clearly means that we experimentalists are still needed. Another another example here is a phosphate transporter where the correction is not predicted. And now what is the next frontier here in structure biology is to get atomic structures in cells or organelles. And you do that in order to find out whether there are differences between the structures which for the isolated protein and in vivo in the natural environment. And here is a very nice example. So people actually went to the slaughterhouse and isolated from pickards the mitochondria. They put the mitochondria entirely on the electron microscope grid, took the photographs, and then they did particle sorting with some kind of tomography in order to classify the proteins, and they could determine the structures of all 4 complexes of the respiratory chain, and this is here complex 1 of the respiratory chain, and the quality is really astonishing. You see here the density for a tryptophan side chain together with the tryptophan, and you even have the lack of density in the center of the ring. Same holds here for the protein. You have even density for water molecules, and you have density in here for a quinone molecule. For quinone molecules, and quinone molecules are here the acceptor of the electrons in complex 1. And so it leads to a much better understanding of what we have, and even new intermediates have been discovered by this in situ crystallography. Next task is localization of each protein in each cell, and for that actually we need immunocytochemistry. We're using antibodies which are labeled in order to find out where are the proteins located in the cell. And this has to be done by many, many proteins. Lastly, I will come to Connectomics, and models of brain disorders have shifted from an emphasis on understanding pathology in specific brain regions to characterizing disturbances of the connections between the neural systems. And so people do connectomics in order to map the neural elements and the interconnections that constitute the brain. The method there are functional magnetic resonance imaging in humans and slicing of brains and electron microscopic investigation of the connections in animals like mice or the fruit fly. And here you see some example of the connectomics, and you see here, for instance, that in the human brain, that these cells up here actually interact with cells down here, and that these neurons here interact with neurons out here. And this is pretty new information, and in order to understand how this works, how our brain works, we have to get more information about that. The most recent progress in connectomics, there is a neuronal wiring diagram of the adult fly brain Drosophila. And this fly brain comprises 139,000 neurons, and more than 50 million synapses have been mapped by an international collaboration. And that's very important to understand that and the changes, because of course flies have memory and they have to make decisions. And with the flies, we might be able to get more information how this happens. The biggest challenges which we still have is understanding our brain, memory, and why do we have to sleep? And most likely we have to sleep in order to fortify the memory. And why and how do we age? And I think for the aging, there is a big recent progress, and I'm personally convinced that aging can be slowed down by a factor of 3 or 4. But I think for me it's too late to get to profit from that. We also have to identify the real cause of Alzheimer's disease and other neurological neurological disorders. And I think we have the methods to answer these questions in our hands, but now we need the young people working in these fields and getting great results. Now I want to turn towards the plant science to agriculture. And that's a field I got a Nobel Prize for work in photosynthesis, and I'm always asked about photosynthesis and the question there is whether we can use photosynthesis in order to supply the energy for mankind, and also in order to reduce global warming. So we try to understand photosynthesis and try to improve the efficiency of photosynthesis. So fossil fuels are derived from photosynthesis, and in photosynthesis plants fix the carbon dioxide from the atmosphere. Now we try to use plants to produce biofuels, to solve the energy problem of mankind, and to reduce global warming by taking carbon dioxide out of the atmosphere. And we have to understand the limitations of photosynthesis, that's basic science, and we can try to improve the efficiency of photosynthesis in a way of applied science. Some basic facts. Photosynthesis, there are two classes of reactions. One is the light reactions. That's the absorption of light, which leads to the creation of chemical energy. Chemical energy here is redox energy and hydrogen, and oxygen. Oxygen is the waste product. With the dark reactions, and here the stored redox energy is used to take carbon dioxide out of the air and fix it as a sugar. And the quantum yield in photosynthesis is close to 100%. That means that each photon absorbed leads to electron transfer across the photosynthetic membrane, but it does not mean that the energy yield is high. First, plants can only use about 47% of the sunlight. They do not use UV light, they do not use green light, and they do not use infrared light. And depending on the wavelength, only between 19 and 33% of the energy of the absorbed photon can be stored in the form of NADPH at the end. And we have a further reduction of the yield by inhibition of photosynthesis at high light intensities, damage at high light intensities, and inefficiency of carbon dioxide fixation. And here I show you an energetic scheme. Here is photosystem 2. Photosystem 2 actually gets excited, and an electron is transported across the membrane. And actually, the electron is replenished from water molecule, and when you take electrons from water, you release at the end oxygen and protons. And the electron is transferred further onto a quinone, a plastoquinone. That's reduced to quinol, and actually, quinol is reoxidized by the cytochrome B6F complex. Electron transfer to plastocyanine. Plastocyanine reduces photosystem 1, and the electron gets energy again by absorbing light, by the light absorption. Then the electron falls down at the end onto NADP, which forms NADPH, which is a form of fixed hydrogen. And the fixed hydrogen is what is used for the fixation of the carbon dioxide. Different way of picture of representation is shown here. So here we have photosystem 2. We have the plastoquinol formation, the reduction there in the B6F complex here, and then the electron flow through the photosystem 1, light-driven electron transfer in photosystem 1 onto NADPH. And actually then we create an electric voltage across the membrane, and we create a pH gradient, and this drives the synthesis of ATP, the universal biological energy currency, which is used for many, many processes. And so we have photo inhibition and photo damage at high light intensities, and this is due to the fact that the electron transfer steps in the later, between the intermediates in the photosystem 2 reaction center is slowed down. So that's very fast from the very beginning, from the primary donor onto a chlorophyll, onto a pheophytin, onto a first quinone, then to a second quinone. And if too much light comes on, actually you get already a second electron before the electron already has left the pheophytin. And then it falls back, and this gap makes problems and can lead to triplet states and formation of reactive oxygen species and lead to oxidative damages. And primarily, one protein called D1 of photosystem 2 is damaged, but the plants have found a mechanism, have worked out a mechanism to exchange this protein 3 times per hour. So very clever by the plants to exchange the damaged protein subunit. And when you look at high light intensities, then it's clear that the photosynthetic efficiency becomes saturated already at a very rather low light intensity. Actually, full sunlight would be here at 1600. So already at 15% of the full sunlight, we have no further increase in carbon dioxide assimilation. This means 85% of the sun energy is lost in that process. And we have limitations also in the dark reactions. We have lack of water quite often. We have an inefficient carbon dioxide fixation. And carbon dioxide fixation happens in the Calvin cycle. So we have a sugar ribulose 1,5-biphosphate, and the enzyme Rubisco, the carboxylase for ribulose 1,5-biphosphate carboxylase, people say Rubisco, take out the carbon dioxide, puts it into the sugar, and the sugar then here is split. And then you also have a cycle with intermediates, and you regenerate ribulose 1,5-biphosphate by consuming ATP, and also an ATPase is used in that process. And Rubisco is a very sluggish enzyme, and it only catalyzes three reaction cycles per second, but the slowness is compensated by the synthesis of enormous amounts. And up to 50% of a mass of soluble proteins consists of Rubisco, and it's the most abundant protein on Earth, and you eat Rubisco each time when you eat some plant stuff. And the problem is that also that it discriminates poorly between carbon dioxide and oxygen by a factor of 80, but the oxygen concentration is much higher than the carbon dioxide concentration. So in every third to fifth reaction cycle, an oxygen is incorporated into the ribulose 1,5-biphosphate. You get an incorrect product called 2-phosphoglycolate, and this is degraded by the plant using a lot of energy. And this energy is about 1/3 of the energy of the absorbed photons for this purpose. So that's really a very project with a great disadvantage. And as a result of these limitations, when you generate biofuels, the efficiency is extremely low. So German biodiesel from rapeseed has an efficiency of 0.08% of the sun's energy, which at the end is stored in the biodiesel. And actually we would have to increase the agricultural land of Germany by a factor of at least 10 in order to produce enough diesel for what we consume in Germany at present. Brazilian bioethanol efficiency is 0.3%. Palm oil diesel is 0.4%. Biogas efficiency in Germany from maize is about 0.2%. And the statement which I came back here, if we use photovoltaic cells, batteries, electric engines, instead of the biofuels, combustion engines, we exploit the energy of the available sunlight better by a factor of 500. And also the use of biogas to generate electricity is by a factor of 100 less efficient than by using photovoltaic cells directly. So that's clear that's the technological solutions are clearly of advantage. And so short discussion about how to improve photosynthesis and what has been achieved. So there has been alternative routes for the degradation of phosphoglycolate. And E. coli, for instance, uses a different pathway. And if you do that, if you replace the phosphoglycolate degradation pathway in the plants by that from E. coli, you get a 50% increase in biomass. And when you try to optimize the Kelvin cycle, and then you also get a 60% increase in biomass, it looks like that there is not enough ribulose 1,5-biphosphate available in order to have this process in a very efficient way. And quite unexpectedly, if you increase the B6F complex concentration in the which leads to a faster oxidation of plastoquinol, then you get less photo damage at photosystem 2, and you get an increase of up to 71% in photosynthetic in biomass production. And these improvements appear to be additive, and there's, in my opinion, a good chance to double or even triple the efficiency of photosynthesis, and which leads to a corresponding increase of biomass and cross yield. And for the moment, of course, we use these techniques in Arabidopsis or tobacco. We have to use that to improve the yield of wheat, rice, and other crops by genetic engineering. And the problem here is that we have to convince the public that this procedure is the best way to ensure that there's enough food for the still increasing world population without the need of further deforestation and reduction of other natural habitats. And of course, an improvement of photosynthesis, more carbon dioxide would be taken out of the air. This also would reduce global warming. And here are the model plants which I used. Arabidopsis thaliana is here, and that's tobacco. So we now have to work to get this knowledge into rice and so on, into wheat and rice and so on. And there is one statement, and more than anything, improving crop photosynthesis in the field will require large-scale collaborations involving expertise from experts in institutes and universities, research centers, and the private sector. And this will require adopting strategies critically assessed as likely to work in field conditions by all stakeholders. And with that message, I want to conclude and thank you very much for your attention.
Thank you, Professor Hartmut. Now I'd like to invite Professor Liu on stage to moderate the Q&A. Professor Liu, please.
Okay, thanks, Professor Michel, for your very insightful and inspiring lecture. Your words really give us a lot to think about. So you mentioned about this, understanding the proteins, how it function. So it's very crucial. And so currently, if we want to understand at the molecular level, what are the tools we can use or the challenge that still exists?
Okay. Of course, in order to understand how such a molecular machine functions, first you have to get the structure. And for the structure you need, when I started as a student, the only way to do that was X-ray crystallography. But for X-ray crystallography, you require crystals. And I had a focus on membrane proteins, and there was the belief that it is impossible to crystallize membrane proteins because they have a hydrophobic surface where they are in the membrane, and they are polar where they are in contact with the water on both sides of the membrane, and there's no way to handle this opposite properties of the protein surface. After about 3 years of thinking about that and working about that, trying various things, I was able to crystallize a photosynthetic reaction center, and we could determine the structure by X-ray crystallography. The alternative methods, which I just mentioned, are NMR, nuclear magnetic resonance spectroscopy, that you can do in solution. It's very it's time-consuming, and it's not a high-throughput method. And the method now, which also is used by us, is single-particle cryo-electron microscopy. And that this method became so powerful is mainly due by the fact that Richard Henderson recognized that when an electron hits a photographic plate, it moves around until it causes the signal. And he thought quite a lot what to do, and he was then able to develop detectors where the signal is already initiated at the place where the electron hits the detector. And this actually led to the increase in resolution. And for the PD oxidase, for our first PD oxidase, it took us 10 years to get the structure. And now with the cryo-electron microscopy, from the start of the project until writing the paper is 3 months. So that is really a huge difference. So we use single particle electron cryomicroscopy for structure determination. And of course, in order to under detection, you try to adjust intermediate stages of the protein, and you study the structures of complexes with substrates and inhibitors, and at the end you might be able to deduce the mechanism of action. Okay. Of course, you involve also theoretical people doing computations. They do MD simulations in order to find out whether you can simulate the activity of the protein.
Both experiment and theory combined together.
Combined.
I'm sure our students also have a lot of questions to share. Maybe this.
Okay, so good morning, Professor. Thank you for your talk. Hi, Mark. I'm from the Philippines. I'm a microbiologist. So you mentioned in the early part of your talk about studying more about the proteins, mostly for humans. But as a microbiologist, I'm interested in microbial systems. And when we do genomic studies in microbial systems, we often get, after annotating, we often get these results of hypothetical proteins. And so my question is, do you think, so hypothetical proteins basically means that they seem to follow the genetic code having an open reading frame, and all of those things, but they don't seem to have known functions, or we don't know if they are really functional proteins at all. So my question is, how do we approach, is it worthwhile studying these hypothetical proteins, and how do we approach studying them?
Because it's clearly worthwhile to identify the function if they have any function. With the membrane proteins, there are a number of small membrane proteins which are just one membrane spanning helix. And what you classically do, you delete the gene and have a look what if there is a function difference. But quite often you find there is no effect of the deletion. Then you still don't know what it is. So we have no clue what many of these hypothetical proteins, they are no longer hypothetical proteins because we can isolate them, purify them. So actually we used mass spectrometry in order to identify these small hydrophobic membrane proteins, but we have no idea what the function is. Yeah? Because the deletion mutants do not show a phenotype. Maybe that we didn't find the right conditions for growing the bacteria. Because the bacteria, they have to react to changes in the environment, and they do that, and we haven't done the correct changes which are needed in order to have a function for that small protein.
Thank you.
Okay, around my right.
Thank you for this tour de force. So I have a question regarding the connectome in the brain. So I would guess that kind of the connectome is heavily influenced regarding the our interaction with our environment. So is there a rough estimation about how much of the connectome is conserved in all humans, how much is influenced by genetics, and how much is influenced just by the interaction with the environment, and how will it make it harder for us to discriminate the connectome between health and disease?
Oh, that is a really interesting question, but this work is just at the beginning, and I do not think that there are answers for these questions. Of course, now with the availability of this fly stuff, yeah. But you have to think that many hundreds of people were involved in doing this kind of work, because in order to identify the location of the neurons, how cells will interact, you need hundreds of people to trace these neurons in the electron microscopic images. We also in Frankfurt we have a colleague who does this in mice, but the work in order to do that under various conditions, I think for the moment is not really doable. Yeah.
Thank you.
Okay, maybe the next one? Yeah.
Hi, I'm Maddie from the University of Melbourne. I'm actually a mass spectrometrist, so this is a super interesting talk to me. Membrane proteins can be quite difficult to isolate and characterize. Do you think native mass spectrometry is a good technique to increase the throughput of characterizing membrane proteins?
I'm sure. Sure. Actually, we had the problems that by doing structure determinations, you quite often identify subunits which had not been discovered by classical means. And for that, we used mass spectrometry in order even to sequence the protein, and we have a limit for 10 kD, which we can simply sequence by tandem mass spectrometry and identify the genes. Yeah? And this is one of the examples I just mentioned. Actually, we have now this protein, maybe 45 amino acids, yeah? And then of course we now have to see the function. So we delete the gene, no effect. The complex is still made, and the function is still the same.
I mean, but if we're doing even a proteomics experiment and we can sequence the protein, it doesn't tell us everything about the structure and the potential function.
It doesn't tell us. We have to still do other experiments.
Yeah, thank you.
Yeah.
Okay, yeah.
Hi, I'm Eric. I'm from Infection Medicine Proteomics lab in Sweden. So every day in kind of clinical proteomics, there are several papers coming out, and currently very few of them have great impact and are able to generalize. And this is really from some fundamental problems, like you have small cohort sizes, very heterogeneous cohorts, but you also have differences in how hospitals and laboratory personnel handle samples,
Yep.
different kind of ways of doing mass spec acquisition types and modes. And you also have different databases, different ways of searching databases. So in the end, you have very little kind of applicability beyond your specific cohort. I wonder what are the kind of steps that has to be taken in order to increase the impact of these clinical findings and actually reaching some real value?
Of course, you need more data. That's quite clear. And so you have to have more and more machines and more powerful machines, in order to analyze that. But I got the impression from our colleagues, in particular Matthias Mann from Martins Reed, who is a world leader in that field, that it's really promising. And what they propagate is, what they propose is sequencing of the blood of the serum proteins, and from the quantification of the serum proteins, you can immediately diagnose diseases. But if you are of opposite opinion, you have to talk to Martin Mann and not to me.
All right, thank you.
You know, these proteins or these three-dimensional, that when they are functioning in the cells, you also have a lot of the environment, like the molecules, sort of the solution, all these, right? So the proteins, the three-dimension, they're also folding. A lot of dynamics here. So whether the current capability that allow us to, monitor all these dynamics at a very, small scale, or, what we can do.
I think structural biology is still will remain small scale. Okay. Will clearly it is speeded up. And one example which I can tell you, when I did the X-ray crystallographic work, in order to collect one data set, it took me about 3 months, because I had a rotating anode in the institute, which I could use, and I had to mount the crystals and to rotate the crystals maybe half a degree, and it took about 8 or 10 hours to do a rotation. So I could do 2 rotations a day, starting one rotation in the morning and the other one in the night, in the evening. And took me about 120 photographs in order to collect one data. You had to scan the photographs and to densitometer them for further analysis. And so it took me one data set. It took me about 3 months. And now, if my students go to the synchrotron, they get the whole thing in 10 seconds in the computer. Yeah?
A big change.
Really, that's a really rapid change. That was a rapid change. But as I said, the limitation still in crystallography is getting enough protein and getting a well-diffracting crystal. And most projects fail on that stage.
I see.
Yeah? But with single particle cryo-electron microscopy, you are much better off. It's much faster, and you can do maybe a data collection for one data set takes now about 4 hours. Yeah? Yeah. And then you can calculate the structures.
Okay.
Calculate the structures.
So the screen is blinking. So we have one last question. Yeah, go please.
Okay, thank you. Good morning, Professor. So thank you for the inspiring talk. So my question is about the brain and the modern lifestyle. So I'm from a background of artificial intelligence, so forgive me if my question is too general. So seems that the modern lifestyle is becoming more and more dopamine driven. So it seems like we are receiving this lifestyle that promotes one single type of neurotransmitter, and it seems like the more dopamine dominates our brain, and the less free will we have. So if we consider the brain as a system of chemical reactions, then shouldn't be balanced by all type of reactions among the materials. So could you share some of your comments or views about the modern dopamine-driven lifestyle, and what did they do to our brain? So in other words, is it taking away the true purpose of our brain? Thank you very much.
The true purpose of our brain, that is a question you have to ask philosophers. I would say that But of course, the brain is everything which helps us to survive. Without our brain, we could not survive. But now that our lifestyle is driven by an increase in dopamine, is really an issue which we have to talk about. Of course, you know that lack of dopamine is the cause of Parkinson's disease. So we would like to have that increase in dopamine receptors in order and also to cure Parkinson's disease, which is very important. But say, for instance, if you are a scientist, you should be curiosity-driven, always to learn new things. And whether this is correlated with an increased dopamine level or not, I would think it's correlated with an increase of the dopamine level when you are successful. But what about when you fail, when your experiments fail? Does this lead to an increase in dopamine or to lowering dopamine level? I don't know. But it's very interesting, your question. Thank you.
Thank you.
Okay, so time is up. Please join me to thank our speaker for this wonderful talk. If you have any questions, you can discuss with Professor Michel during the coffee break. Okay, thanks.